Yeah,
of this
so
chair.
I won't give too much of an introduction because we're actually going to get a very nice introduction, but we have a hometown hero here from one department over who's going to talk about genomics today. And yeah, thanks for everyone for coming. Enjoy. Take it
Well,
away.
thank you. Thanks, David. Thanks for the invitation. I'm very happy to do this and honored, really. I just thought that I would start by giving a very brief account of what my career has been. Like thus far, my PhD is, as David said, it's from Dalhousie, from the Department of Biology, and I got it in '91 in a quantitative evolutionary genetics, or ecological genetics, if you wish. I worked with little fish. And from then I did a short postdoc at McGill. And then as my first son was born, I got no, actually, as my second son was born, I got a postdoc working with Chris Taggart in your own department. And we worked on cod during the time of the post-cod collapse, and we had a very productive period there of 4 or 5 years working together. We're published a bunch of papers on cod population structure and conservation genetics following the cod collapse. Then that led to a job with the Danish Institute of Fisheries Research in Denmark, of course, where I stayed from '98 to 2002 as a senior research scientist in their Fisheries Institute. And in 2002, I came back to Dalhousie as a faculty. So, and I've been here since. So what I want to talk about today is about some research that is a result of some serendipity or some interest that started in 2016, roughly. It wasn't part of my general research program, but it's a topic that interested me, and so I'm going to tell you the story of how this evolved. And it's the topic is, of course, how to use genomics to estimate census size or population abundance in the wild. What happens? Oh, I need to put this into. Sorry. There. So estimated abundance is the first goal in ecology and conservation biology, and certainly in fisheries management. However, estimated abundance with accuracy and precision is often extremely difficult. And we know that from many examples of how difficult it is to estimate how many fish there are out at sea. It's usually done using metrics of catch per unit effort, which requires life history information, and that data is usually obtained from fishery surveys or commercial fisheries. And when we're dealing with marine fishes, with cetaceans or in the terrestrial environments, caribou and things like that, maybe abundance is estimated with aerial surveys. And also with mark recapture of linears. And I presume most people, if everyone in this audience knows about how mark recapture is used, but we'll go briefly over that in a few minutes. The problem with catch per unit effort is that it's subject to biases and uncertainties. If the data span a number of years, they could be gear change, they often misreporting, etc. All sorts of reasons why an estimate of abundance based on catch per unit effort is subject to a lot of uncertainties and potential biases. Air surveys can be imprecise unless the effort is very high. Mark recapture, it's not always feasible, certainly with marine fish. So all of these methods are generally effort intensive, time consuming, and actually quite expensive and often imprecise. So I would argue today that we need a method that if you wish can be a complementary method that addresses these limitations. The method has to be relatively cheap, certainly cheap when we're talking about the value of some commercial fisheries. It must not be subject to tag loss, does not require the recapture of the same individual, and can use dead animals, for instance, from caught through bycatch, should be reasonably precise with reasonably high effort, free of biases, and with the potential to reduce the footprint of research. So the method that I'm going to talk about today is referred to as the closed-king mark recapture, CKMR. CKMR is based on the principle that an individual's genotype can be thought of as the recapture of the genotype of each of its parents. What you do is you sample a large collection of mature individuals of different ages. You capture a large sample of juveniles or younger individuals, not necessarily mature, and you need to age them. And you need to assume that the sampling of the juveniles and of the adults is independent of each other, and that the distribution of these individuals is random. And therefore, if you do that and you genotype those two samples, so those two collections of samples of adult individuals and juveniles, and you use the genetic information to identify parent-offspring pairs, you can interpret that in a mark recapture framework. So the juveniles are a recapture of the genotype of each of their parents. So just to recap or to review what a standard mark recapture thing is you can estimate census size there denoted as NC, as the number, you go in and sample a population one time, you tag those individuals, you put them back, and some time period later, be that a week, a month, or a year later, you go and capture again, that's N2, and you identify the number of recaptures in that N2, then you can use this formula to identify to estimate the census size. For instance, if you have 1,000 individuals, and one, you mark them, you put them back. Sometime later, you go and sample another 1,000 individuals and you recapture 200. That is 200 of those 1,000 individuals bear your tag or bear the mark that you put on them the first time. Then you can go, if 200 individuals were distributed in 1,000, then 1,000 in NC, and you get a population estimate of 5,000. That's the nuts and bolts of the mark recapture function, and it's used in ecology, and it's known as the Lincoln-Pearson estimator, which is it's one of the estimators. It's corrected for biases, and it has an estimated, it has a variance, and that variance can be used to estimate the confidence interval for that estimate. Pretty simple. Now, excuse me, in closed-king mark recapture, you use the same principle, except that the estimate of abundance, NC, in this case has a 2 in the numerator, because each individual has, we're talking about diploid individual, each individual has 2 parents. So here, NJ is the number of juveniles, and A, well, certainly the number of adults. Pop is the number of parent-offspring pairs that you can identify in that sample. And this approach was first proposed by Hans Skog, a statistician at the University of Bergen in Norway in 2001. But back then, this paper more or less went relatively unnoticed, because back then we didn't have the genetic prowess to get enough markers and enough variability to use this with relative confidence. So fast forward to 2016, and this article appeared in Nature Communications, led by Mark Bravington, a fisheries scientist in Tasmania, the CSIRO of Tasmania, where they used this approach to estimate the population abundance of southern bluefin tuna, a highly valuable resource in the Indian Ocean. And lo and behold, they genotyped 13,000 individuals, southern bluefin tuna that were genotyped at 25 microsatellite markers. And they identified, in that sample, they identified 45 and offspring pairs. And that estimate led to an estimate of 2 million individuals of southern bluefin tuna. Paper was published in Nature Communications, and that was my first introduction to this topic. In 2016, I saw this and said, oh, this is really cool, using genetics to estimate population abundance. That paper was followed 2 years later in 2018 by a paper where they used a similar approach to estimate the abundance of white sharks off the coast of Eastern Australia and New Zealand. In this case, they didn't use pair and offspring pairs, they used half-seeds, or individuals that shared at least, were identified as sharing at least one of the two parents. And down there in the slide, I show their effort. They identified, they had 100 juveniles, and they were genotyped at 2,000 plus loci, identified 20 half-seed pairs, and that led to a population estimate of 2,500 to 6,700 individuals of white shark off the coast. So that was really cool. And then that was the same year, now a third paper was published by Robin Waples and the same group, or led by Robin Waples and the same group of Australia, where they looked at the relationship between the genetically effective size, that's a genetic term that if need be I will define later, and the ratio of the effective to the census size. And that was an important finding, because up till then people were thinking that that ratio should be a lot lower in marine fishes than it was actually found to be in this case. So there's these three papers, and I reiterate that I wasn't working on this topic until then, but it interested me. What I was doing at that time was working on some small populations of brook trout here in the Bay of Fundy. Here we have the 14 streams, small streams. They each harbor a small population of brook trout, and I had been sampling them since 2009, and we had data samples from 2009 to 2013. And we were doing back then working with 11 microsats, a decent effort, but not quite high enough. And so this is what we were doing, sampling these populations with electrofishing. And here we see a picture of a colleague in the province in Nova Scotia Department of Fisheries and Agriculture, two of my former honor students helping out. So we sampled these populations over these 4 years, and done the genetics. And what here you have a plot of the results of the genetic composition of this system. And if you focus on this is when all data analyzed together, then you separate this region from these, And the differentiation that you don't see here, and you don't see largely here, you begin to see it as you subset your data set. And then finally, when you have populations individually, you find that most of them are represented by a different color. I should say that each of these vertical bars represents the genotype of a single individual. Different color suggests that the population is genetically different from the neighboring population. And all I want to say here is that these populations are close to gene flow. They're close. You can consider them as individual populations. Gene flow among them is negligible or nil, so the populations can be considered close to immigration. So I had this data when I saw those three papers, the papers on southern bluefin tuna and the white sharks, and I said, hey, this is a very cool thing, which I think we can try to replicate. And the reason I wanted to do that is because in none of these papers, the southern bluefin tuna or white shark papers, there was a comparison with which you could sort of validate your genetic estimates of population and size. How confident are we? I know that the authors of the papers are very confident, but how well does this technique work? And I wanted to try to test that or to validate the method. So we had this, we focused on 4 of those 14 streams, and now we had 7 populations because 3 of these streams have waterfalls. And so we could see that the populations above the waterfall were genetically distinct or distinguishable from the ones below. And so we augmented to increase the number of microsatellite markers to have a better definition, and kept sampling these populations for a number of years. It's another picture of one of the systems, pretty nicely. And here I show you a clip of what it. Of what it means to do this kind of work. The sampling is, of course, non-lethal. We put the fish in a bucket, and then we fin clip them. We cut out a small clip of their dorsal fin and the adipose fin, and then we extract the DNA from those samples. Different pictures, maybe, I don't know why this is going back. So here's a table depicting the sample sizes that we had for the years 2014 to 2018. And you can see that sample sizes are modest. The populations, again, they're not large. The fish are small. And the populations, the streams are in between 4 or 6 kilometers long. And so the population numbers in the hundreds or thousands. And I should say that we estimated population size using double pass electrofishing, which would allow us to estimate the population abundance by the standard mercury capture method. Because the second time we go with the electrofishing machine, we detect which fishes have already been marked and which haven't, or which, by marked I mean which fish is missing their fin clip. And so these are the data we had to work with. And so, but we still had to assign these individuals to age classes. And we did that by the distribution of their size frequency distributions. We knew from other data that a fish that is less than 9 centimeters long is a fish that is 1 year plus. That is to say, that fish is about 14 to 16 months old, and 2 plus and 3 plus. And so these fish don't get to live to 4. So it's a very simple system that we have. Fish that are 1, 2, and 3 year olds, and there are virtually no 4-year-old in the system. And so once we assign fish to their respective size classes, so we ended up with the 58 fish that were collected in 2014, 45 of them were 1 plus, or assigned the 1 plus age class, 2 plus. In this case, we didn't get any 3 plus. And so these represent the numbers of individuals of different ages collected over the period 2014 to 2017. We also had estimates of the survivorship rate, which we had taken from some other data. And this is the survival rate for 1 plus. So 42% of them survive in this stream. 42% survive from age 1 to age 2, only 11 survive from age 2 to age 3, and 0 survive age 3. Numbers differ in different streams. So we now have age, and we now have a survivorship rate. And so these are the data that we went on to continue working. And if we want to sample the juveniles and the adults, well, in this stream, Ross Creek, in the upstream portion of Ross Creek in 2017, we collected 146 fish, and 93 of them were 1 plus. If they were 1 plus in 2017, they would have been hatched. They come out of the gravel in the spring of 2016, and the eggs would have been fertilized in the fall of 2015. So the parents of these 93 juveniles would have been the fish that were collected as 1, 2, and 3 year olds in 2015. So in this case, we have 93 offspring, and the potential parents are here, but not just these guys, but also the ones that were collected the previous years and the following 2 years. And the ones that were collected the following 2 years, only the 2-year-olds and the 3-year-olds would have been present and mature the previous year. And the ones collected in 2017, only the 3-year-olds would have been 1 plus the year of the spawning event. For the fish that were collected in 2014, these would have been present as a 2-year-old in 2015, but not all of them, the ones that got to survive. So you got to weight is 45 by the survival rate. So you don't have to do this if the fish were collected after, if the adults were collected after the spawning event, because if they were alive here, they were certainly alive in 2015. So we did this for the different populations, and so we had age-specific fecundities and age and population-specific survivorship rates for these different populations. So we now are in a position to estimate population abundance using this CKMR approach. But the number of adults, as I hinted already, is not simply the raw number of potential parents' genotypes. That's true only if the adults are collected after the juveniles, and there's no age structure in the population, and every individual has the same fecundity. There is no age-specific fecundity. If that's not the case, and then you need to weight your estimates, your abundance, your adult abundance by the fecundities, the age-specific fecundities, and if the adults were sampled before, by the survivorship. So that formula that I showed before now becomes this more complicated formula, but it's not really that complicated. It's the average fecundity in the population. This is fecundity at age, and it's a relative fecundity, and I'll show you in a minute how you estimate it. And if your samples were collected the year prior or any time prior the actual year of the spawning event, which was 2015, then you have to multiply your numbers by the survivorship at the age they would have been at the year before, in this case, 2014. So essentially that formula becomes this. If we have individuals, this is the year of spawning event, these are my juveniles, these guys, and 2-year-olds collecting 216, you multiply by the fecundity at age 1, and so on and so forth. I don't want to belabor this. But relative fecundity, we know from the literature how many eggs a 1+-year-old produces on average, but only half of them. So 1 year is the age at 50% maturity. So only half of them are mature, and therefore we consider half of this value, 18.4. That's a fecundity of 1, and we compare then fecundities of the 2 or 3-year-olds as relative to that. So a 2-year-old has almost 5 times, is almost 5 times more fecund than a 1-year-old, and a 3-year-old is almost 8 times more fecund than a 1-year-old. So using this data, then we can then go ahead and estimate our population abundance for these 7 populations using the closed-kit recapture approach. And so our goal was to, again, it wasn't specifically to estimate how many fish we have in these small populations, but it was more to validate the approach and compare it to values obtained by the standard mercury capture approach. So, sorry, I don't know what's happening here. I shouldn't use my mouse. But so how did we estimate the mercury capture census size? Well, as I said before, we had we did double pass electrofishing each year. We would go to each stream twice. And the second time, we would count the number of individuals that were missing their fins. And so in 2014, we had 3,855 individuals in RCU and so on and so forth. In 2015, which is a year of that, in this case, interests us, we had these values, and then we had the values later for later years. And we see that the estimates in the different populations range from 400 and the smaller one, small section of one of the rivers downstream of a waterfall, and the largest estimate is around 6,000 fish in this stream. So we're talking about very relatively small populations. So how do these estimates compare with the CKMR estimates? And this is what we came up with. You see that by and large, the estimates are comparable. Maybe in the some of the estimates have wider confidence intervals than the mercury capture estimate. That's not true in all cases. And there was one case where there was no correspondence between the 2 estimates. And U here stands for upper. This is Solnitz Brook, the upper stream, the upper part of the stream, so above the waterfall. So this was eventually, we published this paper in 2019. It had a very nice detailed description of the methodology, which to people like me, at least, it was very obscure and difficult to follow from the succinct Nature Communications or the other 2 papers or 3 papers that had been published. So we thought that we needed to increase the sampling effort to reduce the confidence intervals, but by how much? So here's a detail of the populations that we had. In the first row here, we have 4 populations. The first one was 3,800. The mercury capture estimate for 2015 was 3,800 individuals with a confidence interval that's shown there from 2,700 to 4,900. And we had the sample sizes that we worked in this were 93 offspring that had been genotyped for these 33 microsatellite markers. And we had 214 parents. That together, that's roughly 300 individuals, a bit over 300. We detected, in that system, we detected a median number. We've run this several times. A median number of parent-offspring pairs equal to 7.72. And that, and whereas in this RCD, this downstream portion of that same stream with an estimate of 400, we detected almost 50 plus parent-offspring pairs. So how, the next thing that I want to show is to what degree did we need to increase the sampling effort to get a reasonable estimate of population size, and estimating the coefficient of variation here is one of the square root of the median. And if we want to have a coefficient of variation that is roughly 15%, like here, in this case, we didn't need to increase the sampling effort at all. Just with the 200 that we had here, we were there. In this case, we would have had to genotype a total of 600 individuals. So the increase in effort would have been 100%. So we needed to genotype another 300 individuals for this part of the stream. In this case, and this other one, we only needed to add roughly 75. And in this case, we needed to have yet another 300. So for small populations, the sampling effort is relatively high, but that sampling effort for larger populations, it doesn't increase at the same rate as the population estimate. It's much slower. So the relative effort is lower as the populations increase in size. So this was published in last year in Methods in Ecology and Evolution. And like I said, it was out of the serendipity that occurred as a result of having seen those Nature and the other 2 papers. And there's a mistype there. It's 2015 collections were based on collections taken between 2014 and 2017. So we kept working in this system, and since that publication last year, we just both sampled and genotyped individuals in the following years, 16 and 17, and you could see here the progression of how the populations changed in size between years. In this case, it didn't change so much. Well, in general, there is some variation within the populations. And I should also point out that in some cases, there were disagreements between the mercury capture and the close-kin mercury capture estimates. In here, the following year, we had the opposite effect, and also in here and here. So in both cases, these are the upstream portions of these streams, Woodworth or Saunders Brook. These are streams there in that region of the North Mountain that I pointed out at the beginning. And I have some guesses of why these estimates may not be coincidence or consistent with each other. These are regions that are parts of the streams that are very difficult to reach, and our calculations may have been wrong because the substrate is not always suitable for brook trout. In some cases, it's just sheer granite. And so this is where this work is at the moment. But you know, this is well. Let me just go through the conclusions of that paper. Is that it's possible to estimate census population size using the genetic information if one can estimate the number of parent offspring pairs present in a given sample of juveniles and a sample of potential parents. This is also valid if you are estimated half-seed pairs, HSP. But the requirements are that you have to have age information on your individuals, certainly on your juveniles, and more or less precise age information on your parents. Certainly if the fecundity changes drastically with age, and you have to have some life history information, relatively large sample sizes, and written here in bold, two assumptions that cannot be violated, and that's the sampling has to be random and the individuals have to be mixed. Now there are some limitations for these systems given the relatively high sampling effort required. The method is not likely applicable for very large systems, very large populations, like for instance, if you wanted to do it on herring in the Gulf of St. Lawrence or on capelin, I wouldn't go with these methods because just the number of individuals is just way too many. But it's applicable for marine fishes of certain population sizes. The advantages are no recapture of same individual is required. You can use dead animals from bycatch unless the DNA has degraded, otherwise you can use it. You can read here is no bias from different tag reporting or tag loss, and the precision improves rapidly with sample size. But you need to have information on size or age-specific fecundity. So you will recognize the location of this picture was taken in this July 2019, last year, when we ran a workshop on CKMR right here at Dal in the science building, Ocean Sciences building, and that was funded by a small seed fund by OFI. And you can see Mark Bravington, Robin Whipples, and some of we had 4 instructors, and Joanna here from statistics also participated. So very fun week we had learning the intricacies of these approach. So the goal was to apply this method now that we understand it better and we have a decent understanding of the limitations of the technique, to approach it or apply it rather to harvested marine fish, like for instance Atlantic halibut. As we all know, one of the most valuable, if not the most valuable fishery at the moment. It could also be applied to systems or species that are ecologically important, like predators. For instance, gray seals would be a good system to do it with. Certainly the ones that breed on Sable Island. And I tried getting funded for that, but with no success. Or it can also be applied to species of conservation concerns, like sharks or turtles or something like that. Generally, large animals where the individual is individually valuable and where the population sizes are not in the tens of millions or in the hundreds of millions of individuals. So what about Atlantic halibut? Well, that's where we recently got funded by DFO on a five-year-long project to apply this methodology to Atlantic halibut. And we were working within the context of OFI, one of the modules in the OFI from groundfish fisheries, which incidentally they're having a meeting today and tomorrow. We've got funded to work on Atlantic halibut, and here what I'm showing is the work that is currently under consideration for publication from some of the work that's coming out of that module D, groundfish fisheries. These are collections of halibut from throughout the Northwest Atlantic, from Flemish Cap over here, the Gulf of Maine, southwest Nova Scotia, the Scotian Shelf, Eastern Scotian Shelf, Gulf of St. Lawrence. That's the sampling locations, and there's two manuscripts that are currently, as I said, under consideration. These by Kess et al. It's in ICE's Journal of Marine Sciences, and what it shows is that there's no structure whatsoever within the Scotian Shelf. So if we do it with the Scotian Shelf population, we're certainly doing it with a single population, right? Because one of the things with this method is that you need to know what your population structure is. And we've also, an advantage of doing it with this species is that in the context of that module D, we've also sequenced the entire genome of the halibut, the Atlantic halibut, and we have 86,000 markers, SNPs, distributed among the 24 chromosomes. And so with that data, and Tony Kess, postdoc at DFO, at BIO, led this paper, multi-authored paper, where we find no structure whatsoever throughout the Scotian Shelf in Atlantic halibut. This is a good thing, because then we can estimate the abundance of the population throughout the region without fear of there being different populations in the area. What there is and you see it in these different colors, is that there is some structural differentiation, or some inversions in the chromosomes. There's a lot of variability among individuals, but that variability doesn't show any special structure. So to recap, then, we have the genome sequence of the Atlantic halibut. There are 24 chromosomes, and the genome size is about 734 million base pairs, mega base pairs, and we've identified something in the order of 86,000 SNPs, single nucleotide polymorphisms, that are distributed throughout those 24 chromosomes. If we then assume that all chromosomes are roughly the same size, and each chromosome is about 30 mega base pairs, if we then choose to use 100 SNPs per chromosomes, we went up with a battery of markers consisting of 2,400 SNPs distributed throughout the chromosomes and separated from each other by 300,000 base pairs, which is important so as to be able to assure that these markers are independent of each other. So this is work that is just starting. With COVID and whatnot, even though we knew that DFO was willing to, was going to be approved, the paperwork just was finished in October. But we did collect some samples. So like I said, this is a 5-year-long project. Our goal is to identify no less than 50 parent offspring pairs, and probably a few more. our goal is then to genotype roughly 10,000 to 12,000 individuals. And through simulation work, we think that that will give us roughly 100 and 150 pen-offspring pairs, given what DFO thinks is the population size at the moment, a few million individuals. So our goal is to collect 2,000 individuals per year over the 5-year period, maybe some years more, maybe some years less, and a few hundred through the RV service. And this summer we were able to collect 1,000 or 1,100 fish tissue samples, which are in my office, in my lab, ready to waiting to be genotyped. So this project is in collaboration with DFO, with Melvin Hare, Jonah Mills Fleming and Stats, and two colleagues, Jonathan Fisher and Ole Bree at the Marine Institute in Man. We have PhD students under recruitment. One, I've already recruited one, 2 other ones will be recruited soon, postdoc, one research associate, and summer students that will be involved helping to collect this fish. So I hope this is again, it's a side project which all of a sudden has become a major project in my lab that reflects some interest in something that I found in the literature a couple of years ago, and I said, "Oh, this is good. I think we can do it," and here we are. Other people in the lab are working on other systems. Arctic char in Labrador. Sarah Salisbury about to defend her PhD in the next few months on Arctic char evolutionary biology in Labrador, and Lisette Delgado is working with systems in Patagonia with Galaxia's fishes in Patagonia, also on the evolutionary biology and the genetics of migration. And other students are involved in other projects, including herring and other marine fishes. So with that, I think that I gave you a very brief description of what I'm doing at the moment. Thank you.
Yeah, thanks a lot. That was very interesting. Do we have any questions out there? I thought for sure someone would ask where we could get those great marking recapture t-shirts.
Oh,
the equation on the front, you know, look pretty good.
Yeah, I had them made.
Okay.
I had them made here in it's a t-shirt shop and on Agricola Street, and
Yeah.
you know, that's the second one I had. They always ask me what they mean.
Look good. Well, I'm not seeing anyone jump forward to the questions right away here. May have, you know, but it was yeah, learned a lot about populations and animals. Not our usual fare here, so really appreciate it.
Yeah, no, I know. I realize it's not the usual oceanography subject, but some relation to it.
Yeah, it was very interesting. So thanks again.
Okay.